So let's talk about the evaluations first. You've got, each group has 4 plates in the incubator right now from your transformation activity last time. You got to take a look at those. You've got 2 P- plates, 1 TSA, 1 TSA with can, and you got 2 P+ plates, 1 TSA and then 1 TSA kanamycin plate there. Which of those plates are you going to be counting colonies? If successful, if you got transformants, which of those plates will have colonies that you need to count to represent your transformants, your cells that were transformed by the plasmid? TSA, kanamycin, P+. Those should have colonies on them if you were successful, if your transformation was successful. So let's look at, I pulled the groups plates out. Let's take a look at that real quick. You can see here are the two on the top right here, the 2 P- plates, right? This one is the TSA only, and this is the TSA kanamycin plate. Is that what you expected? Is that correct? A lawn here, no growth here. Is that correct? Why? Explain why that is what you would expect to see. Why isn't there any growth on the kanamycin plate? There's no transformants, so all the cells remain sensitive to kanamycin, and so nothing grew. And then you've got a lawn here on the TSA P+ plate because nothing was selected for. But then with kanamycin, it's selected for transformants, right? And so you can see this one's easy count. 1, 2, 3, 4, 5 colonies representing 5 transformed cells or 5 transformants. So now you need to calculate transformation efficiency. By the way, this was one of my classes yesterday. Every group got transformants. You can see all the colors there in that picture. Pretty cool. And then this was the green, the green plate on the left, and then under the UV light on the right. Pretty cool, huh? So now, transformation efficiency. There is a formula for transformation efficiency. It's going to write TE here, TE. And that is the number of transformed cells. So I'm just going to put number of transformants. That's over micrograms of DNA. So this is related to the how much plasmid you added to your P+ transformation tube. And then times volume at recovery over volume plated. All right, so for group 2, I counted 5 colonies. So that's going to be the number of transformants. That's 5. We need to figure out what the micrograms of DNA is. Volume at recovery. Well, the recovery step, if you recall, was where you added some extra nutrient broth. This was after heat shock, right? After heat shock, you added some extra nutrient broth, and then you put the tubes in the 37-degree water bath for 5 minutes. That was the recovery step. What was the volume of those tubes in when you put them in the 37-degree water bath? If you were to look back in the protocol or your notes, you would see that you had 250 microliters of calcium chloride, and then you added an additional 250 microliters of nutrient broth at the recovery step. So that made the recovery volume 500 microliters. And then how much of that did you plate? It says 50 up there, but if you recall, I had you make a change. You plated 100 microliters. Okay, so we've got everything we need except for that number right there, micrograms of DNA. But if you look in the lab manual or the quick reference guide, it tells you what the concentration of the plasmid was that you used, 0.2 micrograms per microliter, and you added, remember, the tiny 10 microliters to the calcium chloride E. coli cells, right, the competent cells. So you multiply the volume times the concentration. So it would be 10 microliters. I'm going to put it over 1 because I'm going to set it up like a dimensional analysis problem. And then it's 0.2 micrograms per microliter. So the microliters cancel. 10 times 0.2 is just 2 micrograms of DNA. So that's how much total DNA went into that P plus transformation tube. Okay, so now we plug 2 in over here, and then you just do the math. 5 divided by 2, that's 2.5. 50 divided by 100, that's 5. Right? So 2.5 times 5, now we're back to 5. So that's 5 transformants per microgram of DNA. Okay, so that would be your transformation